Hello, everyone. Welcome back to another week of biology. We are at week 5 this week, I think, and we are approaching our halfway point of the course. I know things are getting busy right now for you guys in all the subjects. You probably have some kind of major test or quiz coming up, if not this week, in the next week or so for sure. I just want to wish everyone good luck and that you are able to pace yourself properly so that you don't feel overwhelmed. So let's continue with lecture 4, which is about transport across cell membrane. And at the end of last class, we talked about producing ATP as a way to store energy temporarily, and then we can use that energy for various activities in the cells, including moving certain components. We call those mechanical works. Um, we can do transport with the help of energy if we are going against is concentration gradient, and we can also use them for chemical reactions. So before we go into more details about those scenarios, I want to start with a note today, which is on the topic of tonicity. So please take out a piece of paper, and we're gonna look start with that, okay? So what is tonicity? Tonicity basically is a word that describes capability, the capability of a solution. Solution is like water with some solute dissolved in it, right? It's a capability of a solution to modify, to change the volume of cells by altering, by changing their water content. So depending on what kind of solution the cell is exposed to, it might get bigger, it might get smaller, it might experience no change. And the results of that would be the cell getting bigger or smaller. Uh, uh, the volume might change, and that is what we call tonicity. Okay. So I'm gonna show you 3 scenarios, and I'm going to tell you how a plant cell would behave in these scenarios and how an animal cell will behave in these scenarios as well. So let's start with case number 1. Case number 1. It is the easiest to understand. It is called an isotonic solution. Isotonic solution. Okay, so I'm gonna have a beaker. I'm gonna put a beaker right here. It's like a glassware. Okay, and then I'm gonna put an animal cell in there. Okay, this is an animal cell. This is obviously not to scale. Okay, if you put an animal cell in a beaker, you won't be able to see it. Okay, it's like really small, right? It's microscopic, right? But anyways, here it is, an animal cell. And I am going to do the same thing with a plant cell. Now, plant cells, they have a cell wall, so they are more rectangular in shape compared to the animal cell. Okay, so there we go, plant cell. All right, so do a decent size diagram. All right, so it's not going to be like super crowded. There you go. Now I'm gonna use purple dots to represent the concentration of the solution as well as inside the cell. Basically, the more dots you see, the higher the concentration. So in an isotonic solution, the concentration of the solution is the same as the concentration of the cell. Okay, you know what? Let's take away the nucleus there. Um, Sorry, I just realized it's too big here. Okay, so I'm just gonna redraw my plant cell. Sorry, there's the plant cell. Of course, there's the nucleus inside, but I'm gonna take it away to make more room. Sorry, should have thought of that earlier. There we go. Okay, so it's not about the number of dots, okay? It's about the concentration. And concentration is um, the amount of solute, which is like salt, divided by the volume of the solution. So yes, there are more dots on the outside, but there are also more liquid on the outside, right? So if you work it out, it's about the same outside as it is inside the cell in both of these cases. Okay, so what is an isotonic solution? An isotonic solution is basically when the concentration of the solution, okay, square bracket means concentration, okay? Concentration. When the concentration of the solution is equal to the concentration of the cytoplasm. Okay? The inside and the outside are basically the same. Right. Now we talked about that U-shaped tube last class, right? I don't know if you remember that, uh, but let me just bring it up for a second, right? If the concentration are uneven, then the water will shift towards the side that is more concentrated, right? Water is attracted to the side that has more salt in it. That's why we salt the food, right? To remove the moisture from them. Okay, so that's what we are talking about here. The concentration on the outside and the inside are the same, so there's no uneven pull on water, meaning water will be coming in, but it'll be leaving at the same rate as well. Okay, so water, the rate of water coming into the cell is going to equal to the rate of water leaving the cell. Rate of water entering the cell equals to the rate of water leaving. It's not that there are no water movement, it's just that they cancel each other. So overall, no net water movement. Right? And no change in volume. Okay? No change in cell volume. Yeah, so to visualize this, you can think of a bucket, a basin with a hole at the bottom. And then we're gonna have a tap here. And let's say the basin already had some water in it to begin with. So if you have a hole at the bottom, then it's gonna start leaking out, right? But you also turn on the tap, so the water is coming in at the same rate as the water is leaking. And what's gonna happen to your water level? Well, it's gonna be no change, right? Because you're gaining as much water as you're losing. So they cancel each other, they balance out, no change in volume. Okay, so that's basically what's happening in this case. Okay, just gonna move this stuff a little bit to the right. And so after, this is before, and then this is after. Okay, before. Sorry, I haven't done this note for a few months, so my spacing is a little bit off. This is before, and then this is after. So what's going to happen after? Nothing. Nothing's going to happen. The cell would just be like before. Okay. Animal cell on top, plant cell at the bottom. It's not going to get bigger. It's not going to get smaller. Okay. It's just going to be exactly the way it was. Okay. So no change. No change. So our animal cells, it'll be happy. It likes it isotonic. Okay? All the cells in our body are functioning best when it's isotonic, when the environment is isotonic. For the plant cells, they are okay. They're not like super happy. They're not sad. They're like okay. All right. So not the best for the plant, but it's okay. It's it's tolerable. So that's isotonic. Any questions about isotonic before I teach you the harder two scenarios? This is like the easiest one. All right, moving on. Number two. Case number two. Case number two is doesn't really matter. Let's do hypotonic. Okay, I want you to write the O really big, okay? Hypotonic. Okay? Hypotonic solution. Okay? This one was isotonic, and now we have hypotonic. Hypotonic. So let's do our animal cell again. There we go. In our lovely beaker. Animal cells don't have a cell wall, so they look more like a ball. And we're going to draw the plant cell underneath. Again, for those of you joining a little bit late, these are not to scale. Okay, if you put a cell in a beaker, you will definitely not be able to see it with your eyes. Plant cell. Okay. So hypotonic solution, what does it mean? In a hypotonic solution, the inside of the cell has a higher concentration than the outside. Okay. So concentration square bracket means concentration of the solution is less than concentration of the cytoplasm. All right. So before I tell you the answer, I want you to think about it. I want you to think about the water movement. Do you have more water coming in or more water leaving the cell? What's going to happen?
Can you repeat?
The question?
Yeah.
Yeah. Do you think more water will be coming into the cell or more water will be leaving the cell? That's the question.
Coming, leaking from the cell.
That's correct. Remember, water will always move, if it can, okay, it will always move towards the location that has more salt, okay, more concentration. And that's exactly what happens here. Because of the higher concentration inside the cell, you will have more water coming in than leaving. Right? Water is still leaving. It's not as much. Right? Coming in like this. Does anybody have any question? Rates of water entering the cell is greater than. You shouldn't memorize these things, okay? It should make sense to you. And if it does not, ask me questions. Does everybody understand why more water is going in than leaving? Okay, so if we draw the basin to help you visualize this, 50%, you mean you only understand half of it?
Yeah, I mean, I cannot say I understood 100%.
Okay, do you have a specific question about which part that
I mean, I didn't understand that part. Why? I mean, the water is leaving less than, you
Hmm.
know,
Yeah, sure, no problem. Okay.
coming. Yeah.
Yeah. So, let's just backtrack a little bit. I guess it has been a week, right? So it all comes back to this example here, right? If I have a membrane that only allows water to move through, and the red dots, the purple dots, the dots cannot move. Only water can move through. You have to understand water will always move towards the end that has more dots. More dots means higher concentration, right? This is low concentration. Water will always move towards the saltier end. You might remember I said this last class, right? Put a spoon of salt in your mouth and hold it, right? And after about 30 seconds, you will start salivating, right? You will start drooling, watery mouth, right? Can you visualize that, guys? And that's what's happening. The salt in a spoon, and you hold it, is essentially sucking the water out of your mouth, right? The water is pulled towards the saltier location. And that is what we are experiencing here. If you put a cell in a solution that is less concentrated, it has less salt in the surrounding, then the water will be attracted into the cell because that's where the solutes are. That's where it is saltier, right? It doesn't mean water is not leaving the cell, but it means more is going in than leaving. So overall, the cell is going to be expanding, gaining water. Is that a little bit better? Leanne, you okay with that? Or you have follow-up question or anyone else? Yeah. All right. So it's like this, right? It's like we have the basin again, and this time, it's leaking like before, but we're gonna crank up the faucet, and then it's gonna be like lots of water coming in. Right? So you have a lot of water coming in, and not a lot leaving. And what's gonna happen to your water level? Well, common sense should tell you the water level will increase, right? And eventually it will overflow. Water will spill. Right? And that's what's happening with our cell here. And I keep on forgetting to leave some space. I'm so sorry. So what's going to be happening in this case is the net movement. Okay? Net movement of water into the cell. So our cells, if it's an animal cell, it's gonna be like a balloon that's being inflated, and at some point it's gonna be too big, and then it's gonna pop, right? So the cell is gonna burst, basically, gonna pop. Right? So we call this lysis or burst. Right? If it pops, then of course it's dead. So not happy, no good. There's no coming back. It's dead. Right? Remember, animal cells are most happy when it's isotonic in the environment, and then it's not happy. It's dead if it's hypotonic. Why I asked you to draw a big O is to help you to remember the cell gets really big and it could pop. All right. But if it's a plant cell, then they are actually quite happy. They love this. Okay. What makes it different for a plant cell compared to an animal cell? What do they have that we don't have? And it protects them from bursting.
Centriole.
I think you might be thinking of something else.
Yeah,
Yeah.
yeah. Another one. Valva.
Yeah, they have the cell wall, good. And they also have the central vacuole, right? I think that's
Yeah,
what you
yeah.
were trying. Yeah. So the cell wall, which I didn't really draw over here, right? We draw that in to emphasize it. The cell wall is going to protect the cell from bursting. And also, remember they have a huge central vacuole inside, right? That's able to store all the water in the cell, and it's actually quite happy. Okay, so this is going to create what's called uh, turgor pressure, and it allows your plant to basically, I spelled that wrong, sorry, turgor pressure, which allows the plant to stand up. Okay, so if you water your plant, then it's gonna be able to stand up, and all the leaves are gonna be open, and it's happy. It likes water. Okay, so happy. Right? Whereas over here, this is like when you forget to water your plants for a few days, and then you see the leaves kind of shrivel a little bit, maybe a little bit like sad like this, or they might even be like withering up like that. But as soon as you give it water, then it will open up again because the water gives it a hypotonic environment. Okay, any questions for hypotonic before I tell you the last case? Iso, hypo.
Hypertonic.
That's right. The last one is hypertonic. Okay, hyper. Number 3. Hypertonic. All right. So what's going on with hypertonic? Well, we're gonna draw our nice beaker right here. We're gonna put the cell in like before, animal cell. You cannot see a cell without a microscope. So this is just an illustration. There is our plant cell again. So in the case of a hypertonic solution, what's going to be happening is that you will have more solute in the surrounding than the inside. Okay? Very salty on the outside, like the seawater. Like that. And then just a few on the inside, okay? There you go. So concentration of the solution, square bracket means concentration. It's the official symbol for concentration. It's not something I made up. In this case, is greater than the cytoplasm. Okay, did I get it right last time? I did. Okay, so the solution is greater than concentration of solution is greater than the concentration of the cytoplasm. So think about it again. Which way is water going to move? You don't have to tell me. You just think about it in your head, right? Like, is it going to move more moving out or more moving in, right? Remember, water is always attracted to the saltier location.
Moving out from the cell and central.
Yeah, exactly, right? So again, it doesn't mean water is not coming in. They are, but you just have more leaving. Okay, just have more leaving. Right? It's leaving the cell. So over here we have our analogy to help you understand. Okay. You don't really need to draw this if you already understand it, but I think some people find it helpful. Okay, so we have the basin, but this time we have a really big leak. A really big leak, and all the water's just like going all over the place. And then you don't turn on the tap too much. Okay. So rate of water entering the cell is less than rate of water leaving. Okay. So over time, the water level will drop. Right? Water level is going to drop. So net water, what did I say before? I try to keep it in the same language, but I keep on changing it. What did I say? Net movement of water into the cell. So here we'll say net movement of water out of the cell. Okay, so what's going to happen to the cell? Is it going to get big? Is it going to get small? Well, I'm sure you,
More.
yeah, understand it's going to get small, right? So let's draw what it looks like. Man, I never have enough space for these things. Well, there we go. All right, so this is our animal cell, and it's gonna shrivel up like a raisin. Okay, we have a term for this. It is called plasmolysis. Plasmolysis, shrinking. And this is all right for us, right? Like it's not the best, but we will live. Okay. In fact, you have a part in the brain called the hypothalamus. We'll learn that next semester. The hypothalamus will shrink in response to dehydration. And as a result, it makes you feel thirsty. Okay. So this is plasmolysis. For the plant cell, for the plant cell, uh, it is no good for them. Okay, they're not happy. Okay, this also called plasmolysis. They are not good. The cell wall will still be like that. Okay, but then the cell would like shrink away and become like really small on the inside. They will pull away from the cell wall and basically be very, very tiny. Right, so this is when you forget to water your plant for like a week or so, 2 weeks. Gonna die soon. Yeah, remember, plants need the water to make their own food, right? So those are the three kinds of environment. Okay. Isotonic, hypotonic, and hypertonic. Are we good? Any questions? Okay, let's take a look at our PowerPoint slides, right? In this picture, you can see basically is what we're trying to draw, right? And you can see isotonic. This is a red blood cell. It's fine. It has that very distinct indentation shape in the middle, and it is in the best possible condition that it can be. If you drink too much water and you lose too much electrolytes, then the environment becomes hypotonic and the cell is going to swell up. It might not burst right away, but if it continues to be in this state, then it's going to pop. Over here is dehydration, right? You can see it shrivels up, right? They are going to be fine for a little bit as soon as you rehydrate quickly. Otherwise, it is going to be, causing you to lose fluid from other parts of the body, and that could be dangerous. Okay, if you are dehydrated for too long, your blood pressure might start to drop, and that could lead to all kinds of problems. So if you're gonna become a healthcare provider working as a nurse or practical nurses, you might come into situations where you have to do IV solutions for your patients, right? So IVs are intravenous solutions. And for healthy patients, IVs are usually isotonic to the cell. Okay. You don't want to be giving solutions that are too salty or too diluted and feed it into the person's bloodstream because it will cause their cells to either pop or shrink, right? You want to give them isotonic solution. Right, so that's like the most common type of solution. It's normal saline. That's 0.9%, which is what that means is 0.9 grams of sodium chloride per 100 milliliters of fluid. Okay, and it's mostly used for rehydration purposes after people suffer from blood loss, vomiting, or diarrhea. Okay, so rehydration purposes. Now, in some cases, you might need to have hypotonic solution for certain conditions. Yeah, so you can see the hypo meaning is lower, right? Hypo is half the concentration of normal saline, and this is used to treat situations such as hypernatremia. That's a big word. Emia means in the blood. All right, and then natri is sodium, right? That's why the symbol for sodium is Na because the Latin name is natrium, and hyper is too much. So if a person is severely dehydrated, then they basically have too much sodium in the blood. The blood is too concentrated, in other words. So you need to flush the blood, you need to dilute the blood, and you give them hypotonic saline. All right. It's possible for people to have diabetic ketoacidosis, too much acid in the blood caused by a buildup of these things called ketones. Okay, you'll learn about ketones in chemistry later. All right, hypertonic saline is very rare. My sister is a nurse in her, 15, 16 years working as a nurse. She had never administered hypertonic saline before I asked her. And it's being used in some cases to treat hyponatremia. If a person is, very low in electrolytes, very low in sodium in the blood, they might give them hypertonic salines. It's also used to treat some kind of pressure building up in the brain, apparently, but it's very rare. Okay, so there just like a story I remember. This happened a long time ago. Okay, like more than 10 years ago for sure. Do you guys know that there is I guess there was like Nintendo, they came out with this video game console called the Wii, right? You might have probably know that, right? It's like you play with the movement of your hands on the controller. Anyways, if you don't know, doesn't matter. This is a new video game system that was very popular when it came out 10, 12 years ago. Okay. So there was a radio station in the States. They host a game contest. Okay. The title of the contest was, and I'm not making this up, you can look this up. It's called Hold Your Wii for a we. Okay, so the contestants have to drink as much water as possible and not go to the bathroom. And whoever can drink the most and hold it for the longest will win a free game system. Okay, and so people, went and signed up and participated in the contest. And this lady, she won. She was able to drink a lot of water and not go to the bathroom, and then she won the video game system. And then later on that afternoon, she was found dead in her apartment. So why? How did she die? Based on what we learned today, what caused her to die? Anybody?
Hypotonic cell burst out.
Yeah, basically, exactly, right? So by drinking so much water all at once, all the water got absorbed and goes into the blood. It dilutes the blood, right? It causes your blood to be hypotonic, right? It dilutes your electrolytes, and you basically have something called water toxicity. Okay, too much water, and then the cell starts to burst, right? With the electrolytes going so diluted, you need those electrolytes. The chlorine, the sodium, the magnesium, all those things. You need it for normal bodily functions, right? Including the beating of the heart, firing of the neurons, all those things are not going to be working if the electrolyte concentration are too low, right? And that's how the person died. Very unfortunate. Okay. You can look up that story if you want. All right, and now we are moving on to the last part of our PowerPoint slides. There are a lot of stuff going on here, and I think I'm just gonna make you a note. I think that's always better for the most part than going through the slides. You can read the slides later, okay? It's the similar kind of information, but I always like to draw the notes out. It keeps you from falling asleep too, right? I think. But if you don't like note taking, then that's too bad, I guess. You you can just sit back and watch. All right. So what are we gonna do with the note? I just want to give you a heads up so you don't run out of space. This is a big note, okay? This is your piece of paper. I'm gonna give you a gigantic flow chart. We're gonna start in the middle here, and then I'm gonna go over here, right? Over here, and then under this category, I'm gonna have like 3 categories and then some more categories. And over here, we're gonna have categories, and then categories. Okay? So we're gonna have just like branch all over the place, at least 4 levels in total, and then it's gonna be like going left and right. So plan your space accordingly, guys. All right, let's get back to it then. We're going to start at the middle, and we're going to have the word. Is this not the same chart in the PowerPoint? Okay, the PowerPoint is like a condensed version. I'm going to make it like super big for you with all the information. So here we go. Transport. Transport. Yeah, actually, I can write it bigger. Transport. Okay, and of course, I'm talking about transport across the cell membrane. We're going to try to move things into or out of the cell. And there are different ways to do it, depending on what you are trying to move across and depending on the relative concentration of the things you're trying to move across. Okay, so we're going to have 2 main category on the left here. I'm going to do passive transport first. Passive transport. Okay, just stop me if I'm going too fast. Okay, just say, hold on a second. I'm still copying. It's totally fine. So for passive transport, basically it involves going from high concentration to low concentration. Okay, now I know people can get a little bit confused here because in the case of the water, I said it would always go towards the saltier end, right? But now we're not focusing on the water. Okay, we're focusing on the red dots or the purple dots, right? We're saying if the red dots and the purple dots can move across the membrane, where will they go? Okay, so they would naturally go from high to low concentration, and that requires no ATP from the cell. Okay, this is called passive transport. Okay, think about going downhill. You got a ball at the top of a ramp of some kind of slope. You let go, then the ball automatically rolls down the hill from high to low. No energy is required. Like pushing it up would require energy, right? But no, this is going downhill. So that is passive transport. Okay. Now for passive transport, there are different kinds of examples. So we can start with simple diffusion. Okay, so we're gonna have three examples here. I don't know how you're gonna divide up the space. I'll try my best here. Let's start with over here. We have simple diffusion. Remember last class I showed you diffusion, with the flask, and you drop some blue dyes into the flask, it automatically spreads out, right? That's an example of simple diffusion. Okay, so small nonpolar molecules can cross the cell membrane this way. Okay, so I can give you an example. Right? Something that's small would be like O2 and CO2. These are small. If you have a steroid, these are large, but then they are nonpolar. Remember, the phospholipid bilayer, the cell membrane, is primarily nonpolar in the middle, right? So anything that's nonpolar is going to be able to go through no problem. Okay, steroid hormone, for example. Okay, now I can draw this out for you. Basically, we have our phospholipid bilayer here. There you go. Sorry. Okay, you understand this is the cell membrane, right? We did it last time. So you might have some molecules here on the outside, and then you might have not as much on the inside. And so they will go down their concentration gradient. They will go from high to low. High concentration to low concentration, provided that they're small. It doesn't have to be coming in. They could be going the other way around. You might have these ones, the orange ones, but this time you have more on the inside and less outside, and they will be able to go out. Okay, high to low. Okay? So the movement of the blue ones are independent of the movement of the orange one. They don't have to be moving in the same direction. They could be moving in opposite direction, and that's totally fine. It's every single one is gonna have their own high to low. We call it gradient. Gradient just means difference. Okay, and they'll follow their own paths. That is simple diffusion. Okay, only small things or nonpolar things can do it this way. Okay, so that's passive transport. No energy is required, no ATP, it just happens. Okay. Second one is what we were doing for the past half an hour, this is just osmosis. Okay, so osmosis is nothing fancy. Osmosis is basically diffusion of water across the membrane. Okay, and remember, so I mean, this is obviously just water. And remember, water is attracted to higher solute location. Okay? It will go towards the end that has more salt. I keep on saying salt because it's easier for you to relate, but it's not just salt, right? It's like stuff that are dissolved in water, right? So whichever location has more stuff dissolved in it, water is going to be attracted to it. All right, number three. Number three, we have facilitated diffusion. Facilitated diffusion. So facilitated diffusion, I'm going to put a definition on the side here. It is movement of molecules across the membrane, the cell membrane, with the help of a membrane protein. We learned about membrane proteins last class. Right? In fact, there are 6 functions, right? Attachment to cytoskeleton, intercellular junction, cell-cell recognition, so on and so forth. And one of the functions is to participate in transport. So we call this facilitated diffusion. To facilitate is to help, right? So this is diffusion with the help of a membrane protein. And usually it is for large or charged particles. Okay? If it's charged, it cannot pass the cell membrane on its own. If it's big, it cannot cross the cell membrane on its own either. And so there are 2 types of facilitated diffusion. Let's do this one first. This is called a channel protein. A channel protein. I'll describe it to you. So channel protein is used by charged particles, things that are positive or negative. Okay. The membrane proteins creates a channel or a tunnel, okay, for the charged particle to pass through. An example of this would be calcium ions. Okay, so let's draw this guy out. Now this would be a kind of integral protein where it spans all the way through. Okay, but then it has a tunnel in the middle. Has a tunnel in the middle. And then, if you have more on the outside than inside, right, then they're going to come in. Right? Again, this is still passive transport. No energy is required. It's going from high to low. Right? This is high outside, high concentration. This is low concentration. If the situation switches, like if it's low on the outside and high on the inside, then they're just gonna move out. Okay. Because they are charged particles, they cannot go through the phospholipid bilayer. The hydrophobic tails does not interact well with the charged particles, right? And so by having these channels, it creates a tunnel to shield the charged particles from the hydrophobic surroundings, and then they're going to be able to go through, okay? Am I making sense here, guys? Or you're with me? I feel like it's a lot of information. Any questions? All right. So, what is the other kind of facilitated diffusion? Well, the other kind of facilitated diffusion also involves some kind of membrane proteins. And this one is called a carrier protein. Carrier protein. Okay. So used by large molecules to cross the membrane. Okay, so these are really big, really bulky things. They won't fit through the cell membrane on its own. Okay, so example would be like glucose. That's relatively big and cannot go through. And let me draw it out for you first, and then hopefully it'll make sense. Okay, hopefully you have colors. Otherwise, it's not gonna look too good. Right, so I'm gonna put a carrier protein here. Now just watch first, and then I'll give you some time to copy, okay? Here's our carrier protein, and let's say we have a bunch of these triangle things that we need to move through, okay? I'll draw this in a bit, okay? Just leave it alone for now. Okay, so this triangle thing will bind to the carrier protein. Of course, the shape has to match. And then the carrier protein, which is this guy, carrier protein, it's going to shift the shape, changes its conformation, and now is going to be facing inside, right? And then the thing is going to come out, right? So the channel protein just sits there, no change in shape. Right? Things just go through, right? But for the carrier protein, it actually changes the shape.
Like receptor?
Yes and no. I'm gonna use this as an analogy. This is like those doors you go through to go into the mall, right? Like you walk in one side and then you push the door and then you are inside next. So the revolving door, it's like the carrier protein, right? It changes shape, right? Whereas the channel protein is just like it's like a doorway that's always open. And you just walk right through. Okay? I'll write it down for you over here. The carrier protein changes shape. changes shape. And in doing so, it's called translocation. Okay, translocate. Oh, it's just fancy word for move. Okay, translocate the molecule across the membrane. Once again, this is not does not require energy. It's going from high concentration to low concentration. Okay, it's going through the revolving door, either into the cell or out of the cell. Okay, that is it for passive transport. All right. I'll give you a moment to catch up, and then we'll do the active transport in a second. All right. Continuing with our. Did you have a question? Was it empty? I saw you unmute or. Shohag, you have a question? No? Okay. All right. So let's keep going. I hope you still have space. Over here, we're going to have active transport. Okay, so for active transport, it is going from low concentration to high concentration, opposite to what we have here, okay? Low concentration to high concentration. And it requires ATP. Okay, so this is going uphill. Okay, think of a large snowball or something, and you're pushing it up the hill. That's going to require energy. Okay, it's winter, you might take your kids to tobogganing. Right? This is like going up the hill that no one likes. Okay? Coming down, it's fun, it's fast. Right? No energy required. Going up takes some effort. Right? So we're gonna have two kinds. I'm just gonna give you two categories here for active transport. Okay, over here we have something called the protein pump. Protein pump. It's similar to facilitated diffusion. It involves some kind of membrane protein, but this time it uses ATP. Okay, so membrane protein uses ATP to transport molecules or particles. We'll just say particles, probably better. Particles just means things. Against their concentration gradient. Okay. They want to go downhill, but nope, we're gonna force them to go against what they want to do, against the concentration gradient. And I'm gonna give you two examples here. Number one is something called a proton pump. Can someone tell me what a proton is from chemistry class? Like I know, like proton, neutron, electrons, but then like what element is proton?
Hydrogen.
That's right, it is
Yeah.
H plus, okay, hydrogen. All right. So where do we have proton pumps? Don't please don't confuse proton pumps with protein pumps, okay? Protein pump is talking about the membrane proteins, and proton uh is an example of that, okay? So H plus. So here we have, let's see if you can tell from my amazing drawing skills. What organ is this? Can you guys tell?
Stomach.
Oh,
Stomach.
oh, thank you. Okay, that made me feel better. This is the stomach, and of course you know that the stomach
Butyric
can,
acid. Yeah.
yes, the stomach contains acid, right? So we have some stomach acid here, and the acid is a hydrochloric acid. And to maintain the acidity, you actually have a lot of proton pumps along the walls. Okay, and their job is to keep on pumping H plus into the acid. Okay, the more H plus you have, the more acidic it's going to be, right? So you already have a lot of acid, a lot of H plus in the acid. You already have high concentration in the acid, but you want to keep on adding more to it. So that's going from low concentration to high concentration, right? That's why it's an example of active transport. Okay, so people who suffer from heartburn, for example, if they have a lot of acid reflux, they have to take PPI, okay, proton pump inhibitors, right, which basically block these proton pumps and it lowers the acidity of your stomach. I know because I take some of these things, for a few weeks every year. By the time it gets to June-ish, I'm usually a little bit stressed out, and then the acid reflux gets bad, and then I have to take the PPI, so proton pump inhibitors. Another one is called the sodium potassium pump. This is very important for next semester when we learned about neurons and things like that and how they work. I'm just going to give you a brief overview here, just to show you what it looks like. So in your neurons, you're going to have these sodium potassium pumps, and they kind of look like this. Not really, but this is like you know, a cartoonized version of it. So it has two grooves on one side and three grooves on the other side. And on one side, this is where you will fit the sodium. And then on the other side, this is where you will fit the potassium. Okay. So this is the inside. This is the outside. You don't have to memorize the details. Okay. I'm just showing you because, you're going to learn it, uh, a lot more closely next semester. But basically, your cell wants to keep on pumping sodium out of the cell. Okay, three sodium is going to get pumped out in exchange for two potassium. And in the process, you need to have ATP for this to happen. Right? So if you think about it, the sodium keeps on getting moved out. So you basically have low sodium here because you keep on pumping it out. And you have high sodium outside. So you're going against the concentration gradient. Similarly, for the potassium, since you're bringing all of it in, you already have a lot inside. You don't have a lot outside, but you want to keep on bringing it in. So going from low to high, that requires ATP. Okay, so both of these things is an example of a protein pump. It involves some kind of membrane protein, and you're pumping things against their concentration gradient. All right. Almost there. Not quite, but almost. Okay, we're gonna skip the break. We'll be done by 2 probably. Next, we have bulk transport. Bulk transport. Bulk transport is basically moving large quantity of things across the cell membrane. Not necessarily like big stuff. I mean, it could be big stuff, could be bulky things, but it could be a lot of smaller things as well. Right? You're moving large quantity of substance. So let me write down for you. Movement of large quantity or very bulky things. Okay. I say things here instead of particles because it might be like alive things. Okay. Like if you're eating a bacterium or something, then that's like, a living cell, right? Very bulky things across the cell membrane. Okay, so that's bulk transport. And for bulk transport, we have three types. Actually, sorry, we have two types. Let's do this one first. This is called exocytosis. That's right, Naja. Thank you. Exocytosis. The exocytosis is to expel things out of the cell. Okay. Now I'll draw this out for you. Maybe you watch first so that you understand what's going on, and then I'll give you some time to draw. Okay, don't draw this yet, just watch, okay? Because I'm gonna erase things in a bit. This is the cell membrane, but like a lot zoomed out so you don't see the phospholipid bilayer. And then here we might have a vesicle of some sort. Maybe it got repackaged from the cell, from the Golgi body. Okay, and then we want to send this thing out of the cell. That's one of the destinations, right? It can be sent out of the cells, could become part of the cell membrane. It could stay in the cytoplasm. So this guy is going to move towards the cell membrane, okay? And then it's going to basically touch the cell membrane. Then it is going to fuse with the cell membrane. Okay? I just follow the animation here, really low-tech animation. And these things will start to come out. And eventually, right, it will become part of the cell membrane. The vesicle will become part of the cell membrane, and then all the stuff are going to come out of the cell. Okay, so this is exocytosis. It's like expelling things out of the cell. All right? You're not moving one or two of these things out. You're like sending out a bunch of them. And maybe these are hormones, maybe they're, some kind of proteins that you need. Maybe it's like signaling molecules. Doesn't matter. It's a bunch of them sending it out of the cell. Okay, so this is outside. This is inside. Everybody okay? Many of you are very, very quiet. I don't know if you're with me. Hey, let me know if you have any question. All right, the next part is endocytosis. Okay, and endocytosis is the opposite of exocytosis. Instead of expelling things out of the cell, we are bringing things into the cell. large things, okay, bulky things, right? And depending on what you are bringing in or how you're bringing it in, there are three different options. I'm really sorry. This is the last part of the flowchart, okay? I know it's already a lot. But remember, I did not make these things up. I am just the messenger. Okay. So, oh, I gotta restart the program. It's getting a little bit glitchy again. There we go. Okay. We still need to draw the third box right here. There we go. I'll put the names down first and then I'll explain. First one is phagocytosis. And then we have pinocytosis and receptor mediated. Oh, that's really bad. Receptor mediated endocytosis. All right. So what does it mean? Phagocytosis, that's a CY right there. Phagocytosis it literally means cell eating. Cell eating. So for example, if you have some kind of white blood cells here, and then there is a bacterium, then the white blood cell is going to surround its cells around the bacterium, and then like this part and this part is gonna close, and then it will trap the bacterium, and then it eats it. Okay, so this is called phagocytosis, cell eating. Now, if this is cell eating, you can probably guess what this is. If they gotta eat, they gotta drink as well, right? So this is cell drinking. It's what it means. Cell drinking. So it's the same thing, but instead of bringing in some like solid stuff, it's gonna be bringing in mostly fluid. So I can give you an example. A human egg can bring in fluids from its surrounding to obtain nutrients. Okay, so whatever is dissolved in that fluid, it's going to get internalized, and then you're going to be able to use those energy. Okay. Next, we have receptor mediated endocytosis. It's pretty much same as the other two kinds, but with more selectivity. So cell membrane here again, cell membrane right here, and on the surface, you would have special kinds of receptors. Okay, so let's say the surrounding in the outside, you have triangle stuff, and then you have circle stuff, right? Different shapes. Okay, and then you might have like whatever, like rectangle stuff, okay? So the receptors will only allow specific shapes to come in, okay? They only allow particles of a particular conformation, particular shape to come in. So only the triangle will be able to bind, and then it will trigger this portion to like basically forms a vesicle, and then it brings it in just like phagocytosis. So it's like this thing, right? That babies play, the shape sorter, okay? Like you gotta find the right opening to go in. It doesn't just let all the shape goes in. Right? It has to match the opening exactly before it would go into the box. And that's what receptor-mediated endocytosis is like. It's not just going to let anything come in, right? For it to come in, it has to match the shape of the receptor. Otherwise, you stay out. All right. So there you have it. All the different kinds of transport, and everything is in the PowerPoints. Okay. Like I just didn't want to read the slide to you, right? But it's all there. You can read it yourself. Okay. And it should make sense. And that's it for lecture 4. Now I have a few things to say before I let you go. Right. Factors that affects membrane permeability. Right. So the cell membrane, as you know, it's fluid, mosaic, right? It's dynamic, things can shift around, right? And there are different things that could affect the permeability, right? The membrane composition, we already talked about this. If you change the number of percent of cholesterol, percent of saturated or unsaturated phospholipids, then it's going to affect the permeability. It might become more liquid or less liquid, right? You can rewatch the recordings from last class. Temperature might play a role as well, and that's what you're going to be investigating with the lab. Changing the pH, ionic concentration, all these things can affect the membrane permeability. So for your beetroot experiment, you will be investigating the effects of temperature on the membrane permeability of beetroot. There are some questions here, and then you can go through these things yourself, okay? I'll post the answer to them later on. I do want to talk about the lab. Sorry, that's where I got the shape sorter picture. Um, so wrong course here. You are 201. And couple of things happening, right? Wednesday, is it Wednesday? Yeah. Wednesday, no class. Okay, we already finished all of lecture 4. We are good with sorry, this is the wrong class again. Wednesday, no class right here. Okay. You just have to work on your lab. You can in fact start working on the lab today. All right. But you have to hand it in by Wednesday. Okay, you gotta hand it in by Wednesday. It's due midnight, 11:59. You gotta hand it in. And don't no class in the morning. Just take the time to wrap up your labs, okay? Next Monday, you will have quiz 3 and 4. So you can scroll down here and you can see the information on quiz 3 and 4. Sorry, I'm losing my mind here. Quiz 2, which is on lecture 2 and 3. Here are the concept chat answer. The questions we didn't do, like these ones, right? Like we didn't have time to talk about these yet. So you can try them and you can check the answers yourself. Okay. And then quiz 2 will be available next Monday from 6 a.m. to 11:59 p.m. 50 minutes, 25 multiple choice, 2 short answer questions, pretty much like last time. Okay. And then the lab is right here, Beetroot Lab. Okay, so I showed you this last time. I'm going to go through it again briefly with you. This time, I'm going to show it to you through the
One question, can I
Yeah,
ask?
yeah, sure. Go ahead, please.
Quiz two, multiple answer or single
Yeah,
answer?
just single for this time. Yeah. The
Okay,
multiple answers will be for the test. It's going to be coming. Okay.
okay, thank you.
Yeah. Lashante, you have another question?
Yes, hi Vincent.
Hi.
Um, in regards to the Beetroot Lab, can you clarify something for me with question 4 and question 11 once you open,
Yeah,
please?
sure. What is it again? 4 and 11, did you say?
Yes, question 4 and
Yeah, yeah.
question 11.
Sure, no problem. Okay, so this is what you need to do. You need to go into the Beetroot Lab folder and then you click on the Beetroot Lab, okay? You have to start your attempt. I didn't put a time limit on it, so you should be okay. Now, I don't know about the new system, okay? Um, Previously, in another system, whenever you start a quiz or something in one subject, it doesn't allow you to start a quiz in another subject. So I don't know if you have a chemistry quiz or something tomorrow, right? So if you do, then maybe wait until you finish your chem quiz before you start this attempt, okay? Because if you start the Beetroot Lab, which is done on the quiz platform, it might prevent you from starting your other assessments. Okay. I don't know for sure. That's just how it was in the old system. I haven't had a chance to test it out in the new system yet. In any case, you can start it. And like I said, the instructions are all here. And you have to watch the videos before you answer the questions. The videos are not too long. Okay, they're just like 5 minutes tops each. Right. So what is the purpose of this experiment? Here, you just type in the answer, right? I'll type this one for you for free. All right. So the purpose of this, the purpose of this experiment is to investigate, investigate the effects of temperature on membrane permeability of beetroot cells. That's it. Okay, you can copy this one. I give you permission to write exactly like I do here. Right here, I'll even put it in the chat for you, so you don't even have to take a picture. Right here, I'll put it in the chat for you. All right, that's all I'm looking for. All right, nice and short and concise. Don't write me a novel, guys. All right, it's just one point. Answer the question and move on to the next one. And what is the name of the main pigment that creates the distinct vibrant red color in the beetroot? Do not go Google it. The answer is in the video. Just watch the video and get the answer. It's much easier that way. Okay, if you Google it, it's gonna give you some names that I did not mention, and then I will know you Google the answer. All right, don't Google it. Okay, provide 3 reasons why beetroot is suitable specimen. Yeah, so again, all these things are explained in the video. Just watch it. It's quite easy. Was this a question you want clarification on?
Yes.
What's
So
confusing for you?
is it in regards to the beetroot or is it
Yes,
in regards
all
to
the
this
question is
video?
it's in regard to the beetroots, okay? The videos are just there to help you refresh the memory. So like if you forget what dependent, independent variables are, you watch the videos, and then I use another example to help you understand what it is, right? But when you answer it, it's always about the beetroot experiment, okay? So that's what you're gonna type here. The dependent variable in this experiment is da 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 da, and then the other one. Someone asked, all answers are based only on the videos. Most of them, yeah. As you progress through the labs, like there are some questions that requires you to analyze and think about the situation a little bit more. Most of the answers are available either in the lecture notes or some things we talked about in the lectures. Okay. Um, I don't think you need to research anything. Okay. See, it says do not search the internet for answers. Most of it would be all of the answers, actually. Vast majority of the answers will be in the videos, in class notes, or previous lectures. Okay.
One more
Um,
question, sir.
huh? What's that?
One question.
Sure, sure. Yeah.
Safety precautions in terms of the person who is doing the experiment. Yeah.
Yes. So like later on, there's a question about safety, right?
Yes.
So I'm gonna tell you right off the bat where people lose marks. Okay, people are gonna say, "Oh, wear gloves, wear goggles, wear aprons, wear this, wear that. All those things is just one safety precaution, right? It's like wear protective personal protective equipment, right? Okay, so like you really want to think about this a little bit deeper. Okay, standardized variable. If you don't know what it is, then go back to lecture 1. I gave you an example here to help you. Do not use this example in your answer, obviously, right? Some people, forget that and then they use the same thing. Then I will only be able to give you half the points. Okay, so pretty straightforward. Hypothesis. Remember, hypothesis is a tentative answer, right? So you don't have to explain anything. You don't have to explain why you think the temperature will do this, will do that. You just have to state it, okay? Now, people sometimes write the hypothesis like this. Temperature will affect the membrane permeability. Okay, and then they won't get full marks for this. Can someone tell me why? This is a 3-mark question. Okay, why would they not get full marks for this hypothesis? Okay, they might write more to explain what kind of results they expect, but what's missing here in the hypothesis? Someone please tell me. Help the class out. Nothing? This looks okay to you to you guys? Temperature will affect the membrane permeability. Yes, thank you, Samuel. You have to tell me what you think about the effect of it is, right? Like, do you think temperature will increase the membrane permeability, or do you think the temperature will decrease the membrane permeability, right? And so you have to pick a side. You can't just say it will affect it. Like, affect it how, right? And then you have to tell me what kind of results do you expect if your hypothesis is correct. Always pay attention to the points, okay? If it's 3 points, probably you need 3 sentences. Okay, that's a good way to gauge whether your answer is too long or too short. Okay, everything else should be pretty self-explanatory. I don't think there is anything that stands out particularly. 11, you were saying write down your observation. So basically look at the picture and describe what you see. Okay, this is a qualitative analysis. So like I'm not asking for numbers or anything like that. You just describe it using, your senses, right? That's basically what it is.
Okay, so qualitative analysis, are you meaning from what we give you, or what does it actually mean?
This is considered as explain what this means. Yeah, so explain to me what qualitative analysis means. What is qualitative, right? Observations,
Perfect.
right? Yeah, okay,
Okay,
good.
thanks.
Yeah. There is a thing here that's not showing. I'm not sure what it is, but maybe it's not important. It's very glitchy, this thing. Okay, and then there's the calorimeter. You can read it. I already explained. It's me in the video, okay? So like I know it's based on the lab, so it's very tailored to it. So watch it. It will explain to you what the calorimeter does. And then you have to plot the data. And this shows you how to plot the data. And you just have to upload it, okay? So you take a screenshot or you take it from your phone and then you put it on your computer and then you upload it, okay? Like when you try to type your answer, there's an upload button right here, attachment, okay? And then you can choose something from your computer and upload it. Got it? Okay. So make sure you watch the video on how to create the graph. People try to draw the graph by hand in the past, and they're not going to get good grades for it. Make sure your axes are labeled. And also, don't give me a link, okay? I don't want to have to click on a link to take me to your Google Drive or something like that, right? Just take a picture, upload the graph, and it should be okay. Everything else is fine. I do want to do one more questions with you from the PowerPoint slides that will help you answer number 18. But before I do that, does anyone have any questions about what is expected for the lab? No, you do not draw the graph on paper, okay? Do not draw the graph on paper. Someone just has that, okay? You have to plot it on the computer, okay? And if you don't know how to do it, just watch this video, right? You can click on the video and it will go through how to graph it, okay? You can click on the YouTube button to make it bigger. Any other question? It's pretty clear, right? I hope it's 10%. Don't wait until the last minute. You should be okay. All right, so I'm gonna exit this thing. Okay. I'm gonna do this question with you, and then that will be it. After freezing and thawing some animal cells, what would happen if they are placed in a hypotonic solution? So let me do the polls, okay? Just choose what you think. Most people will get it wrong the first time, but don't let that deter you from picking an answer. I'll explain to you what it means after. Go, go, go. Okay, it looks like that's all the answers that are coming in. Well, a lot of people chose the animal cell will swell and burst. And that's not surprising because you see the word hypotonic and then you think about what we did in class and you think, oh, the cell would swell and burst, right? That's what we said in class. But the trick here is that it says after freezing and thawing some cells, right? So when you freeze a cell, what happens is water actually expands as it freezes, right? You know that if you fill up a water bottle to the top and then you put it in the freezer, it actually will expand and sometimes it might even burst, right? Depending on how strong the cap is. And that's what's going to happen when you freeze an animal cells, okay? The ice will expand and then the ice crystal will actually poke holes in the cell membrane. So after you thaw the cell, right? After you thaw the cell is already full of holes from the freezing process, from the ice crystals. And it doesn't matter if you put it in hypotonic, hypertonic, or isotonic, it's just full of holes already. So water will just go in and out without any kind of regulation. So the cells will actually not change in size. Okay, so keep that in mind as you are answering question 18. That is all. Again, don't come on Wednesday. There are no class. Just work on your lab. And if you are run into any particular problems, I can, email me and I'll try to answer you. You cannot send me answers of your labs and ask me to go over it though, right? Like I can only clarify questions for you. I cannot proofread your work. All right. That's it, guys. Have a good one. See you next time, next week, right? Next Monday. Bye-bye.